So our next talk will be by Urna Basu, and she will tell us about direction reversing active Brownian motion. So please.
Thank you very much. Can you hear me? Okay. Yeah. So I'd like to start by thanking the organizers for inviting me here. It's great to be here. So I'm going to talk about direction reversing active Brownian motion. So this is based on some joint works with Sanjeev Sahapandit at RRI and his students Ayan Shatra and Shashank Prakash. So this is the outline of the talk. So I'll give a very brief introduction to active particles. We have already had some talks on active particles. So it will be a brief introduction, and then I'll introduce this particular model, direction reversing active Brownian particle, and then I'll discuss the different dynamical regimes that emerge in this kind of motion, and also some novel scaling distribution that we obtain for this model. And depending on the time, I'll discuss the behavior of this particle in trap and also a little bit about interacting traps. Okay. So yeah, this is the usual active matter introduction. So active matter refers to a collection of self-propelled or active agents, each of which have their own engine. It can convert energy from, take energy at an individual level and convert to directed motion. And because of this, they are inherently non-equilibrium in nature. And examples can be found at all scales, starting from bacteria to bird flocks, and even in labs like micro nanoswimmers or genus particles of various kinds. And the interesting thing is that these particles show not only novel properties not only at collective level but also at individual level. So here I am going to talk about individual active particles. So one attempt from the theoretician side is to use minimal statistical models without going into the details of the chemical cycles in bacteria, but just modeling the motion using statistical models. And there are several models existing, and these have certain common features like which one is the pointer? No? Okay, yeah. So all of them have certain common features. One thing is that they have some internal degree which changes stochastically, and that couples with the positional degree, giving rise to the directional motion. So two of the most famous models are the so-called active Brownian particles. Sabine already talked about it. So here the orientation of the particle changes via slow rotational diffusion, and the other one is the run-and-tumble particles. We heard about it yesterday a bit, and in this case the particle changes orientation in a discrete manner. So it just, the direction of the motion changes in a discrete manner from one angle to the other. And so however, there are certain kind of motion that is observed in nature and in lab where none of these models work very well. So here the first one, yes. So the first panel shows the motion of kind of bacteria called Mysococcus xanthus, where you see that the particles reverse direction after a certain while. And the second panel shows another bacterial motion which is called Pseudomonas putida. It also, if you follow the blue track here, so it also reverses direction after a certain interval, a stochastic interval. And the third one is an experiment done with light switchable titanium oxide genus particles which are half coated with gold. And these also show, okay, just one second. Yeah, a motion which reverses its direction suddenly in an intermittent manner. So none of the, this ABP and RTP models, they do not describe this kind of motion. And there are some other bacteria also, marine and soil bacteria, which show similar kind of motion in the sense that they reverse their direction in an intermittent manner, and between two reversals they show more or less ABP kind of behavior. Okay, so the question is how to understand this kind of motion, or what kind of analytical predictions can we make about the position distribution. So we here introduce a very simple model. Okay, so we have an overdone Brownian particle moving with a constant speed v0 like ordinary active Brownian particle. So the position and the orientation are x, y and theta. We are talking about in 2 dimensions. And so the orientation evolves with the Brownian motion. like in the case of ABP. So we have theta, and theta undergoes a Brownian motion, dr is the diffusion constant. But on top of that, to model the direction reversal, we introduce another variable sigma, which is a dichotomous variable, which can take values plus minus 1, and it can change sign with some rate gamma, so giving rise to the direction reversal. Okay. So we have two different kinds of noise here. One is the usual Brownian motion of the orientation, and then there is the additional sigma variable, which I mean, there is a time scale associated with it, which is gamma inverse. So without any loss of generality, we assume that the particle starts at the origin. So wherever it starts, we call it origin, and with some orientation theta naught. Okay. So between direction reversals, these behave like ordinary ABP particles, and so similar models have been used to understand certain collective behavior numerically, but there has been no analytical understanding beyond just calculating the variance. So our objective here is to see how the non-interacting system behaves. What is the position distribution of a collection of such particles? Okay, so before going into the details, let's have a closer look at the dynamics. So we can recast it in a form which is similar to Brownian motion. However, these noises, these effective noises zeta x and zeta y, are very different. So this is the form. So it involves two components. One is the diffusion, with a time scale dr inverse, and then there is a dichotomous noise sigma t, which has a time scale gamma inverse. So we have two inherent time scales, unlike the usual active particle models, and we expect to see some effect of the interplay of these two time scales in the position distribution also. So here is a typical trajectory at short times. At short times, you see that this looks very different than RTP, ABP, or usual Brownian motion. However, if we look at long time, so this is a very long trajectory, and it's hard to distinguish it from ordinary Brownian motion. Okay, so we already expect some kind of dynamical, different dynamical regimes to emerge if we look at it closely. And so here are some simulations with two different cases. First one is, what happens if the intermittent reversals are more frequent than the diffusion. So when gamma is larger than dr, we have more reversals compared to the diffusion change, and in the other case, reversals are infrequent. And we look at how a cloud of this particle. So let me start it again. Yeah. So we start with 5,000 particles, non-interacting, all at the origin. Whatever point we call the origin, oriented along a certain direction, pi by 4 here, and we see how the cloud evolves as we change time. As time evolves. So in one case, we see that the cloud becomes isotropic quite quickly, and in this case, you see that it retains the information about the initial orientation for quite a long time. Okay, so we sort of expect that depending on whether gamma is smaller or larger than dr, we will have different kind of dynamical behavior, and this is what we are trying to see a bit more rigorously. Okay, so let me summarize what we see, and then I will come to the details. So what we see is that depending on the value of gamma and dr, we can have 4 different dynamical regimes. One is the short time regime, when the observation time is much smaller than both the time scales of the system. And then one is the long time regime, where the time is larger than both the time scales, and here we expect from the trajectory picture some kind of effective diffusive behavior. And then there is an intermediate regime. Yes. And then there is an intermediate regime depending on the order of gamma and dr. So if we are at a time when the time is larger than one time scale but smaller than the other, then we see a completely different behavior, and we will see what it is. But before that, just to give you an idea about what are these time scales in real systems. So for Mysococcus xanthus, gamma inverse is of the order of 100 seconds, whereas dr inverse is 10 to the power 6 seconds, so it's very well separated. And in the other case, for Pseudomonas putida, we have gamma inverse which is 0.1 second, and dr inverse is 40 seconds. So in this case also it's more or less 2 orders separated. Okay, so so this is kind of a summary of the results. So what we see is that at very short time, the distribution is very anisotropic. The particle remembers its initial orientation, and the motion is also very non-diffusive, which is common to all sorts of active particles. And then at a very long time, we have this isotropic, diffusive, and Gaussian kind of behavior. At least the typical fluctuations are Gaussian. On the other hand, the intermediate regime depends very strongly on the order of gamma and dr. So if we have more reversals, I mean gamma is larger than dr, then we actually get a very anisotropic and non-diffusive regime, which is novel, which has not been seen in case of active particles before. And in the other case, when dr is larger, we get a behavior which is again isotropic and Gaussian. Okay. Yeah, so here are some schematic pictures of the distributions in these different cases. So the top panel is for gamma greater than dr, and the bottom panel is for gamma less than dr. And at short time regime, we see that in both cases we get a I mean kind of qualitatively similar picture. So the distribution is like a hammer shaped, it's very anisotropic. There is a peak near x equal to v0 t, I mean v0 is a constant speed, and at very long time we have a Gaussian behavior, single peaked at origin. And in the intermediate time, in one case, we again see a Gaussian like single peaked diffusive distribution. On the other hand, here we see an anisotropic one. So this is the direction parallel to the initial orientation, which is elongated, and perpendicular to that, it's shortened, so it's an oblate shape, and it is, I mean, we'll discuss what exactly this is. Okay. Okay. So yeah. So here is short time regime. I will not go into the details, but just let me point out one thing is that at short time regimes, time is much smaller than both the time scales. So DRT is very small, and then the effective noise takes this form, and if we calculate, we can calculate the mean square displacement here, and it turns out that this is growing as t cube, so it's very non-diffusive. And if you look at the mean square displacement along x and y, they are different, so which means that the motion is also very anisotropic. And if we look at the distribution, this is the shape of the marginal distribution. So this was the 2D full 2D distribution, which is hard to calculate. But if we look at the marginal distribution, there are certain signatures of the non-diffusive, non-trivialness of the motion. So we have a plateau across the origin, and then there is a Gaussian peak. So the Gaussian peak is coming because of the ABP-like behavior. So ABP also has a peak at short time, but now because of the reversals, we get this extra plateau near the origin. So some particles start to move backwards, and they come back to the origin, and they populate this region. Okay, so and the next regime we look at is a very long time regime, where we already expect kind of diffusive behavior, and this is also what we get if we calculate this thing. So here the effective noise becomes like a white noise, but with a different strength. So the effective diffusion constant depends both on dr and gamma, and we get the typical fluctuations which are Gaussian. However, one can ask the question that is there no signature of activity in this regime? The answer is yes. So if we look at systematically the corrections around the Gaussian, that can also be done, and we see that at times which is still larger than both the time scales, but I mean at each order of this tau by t, tau is a characteristic time scale in this regime. We can write the distribution as a power series. So again, I will not go into the details, but these are the shapes of the corrections which are very non-Gaussian. So to leading order, the fluctuations are Gaussian, but then if we look at corrections around the Gaussian, we get very different shapes. And then the third regime, where again, as I showed the 2D pictures here, we again get a single peak Gaussian like distribution, and as expected, if we do the calculations here again, we get an effective white noise and typical Gaussian fluctuations. But now let me come to the most interesting scenario, which is this regime where t is much larger than gamma inverse. So reversals are much more frequent, whereas the diffusion of the direction is not so prominent. So the change in the direction mostly comes from the reversals. So in this case, if we look at the effective noises, then we see that we can write it as a product of a white noise, it is a white noise, and some term which involves just ordinary Brownian motion. And this is exactly what gives rise to this non-trivial behavior, So if we look at the mean square displacement, we see first of all that this is still anisotropic. So the mean square displacement along x and y are still different depending on the initial orientation. Yeah, so first of all, I mean, to leading order it looks like diffusive, but if cos 2 theta naught is equal to 1, then there is a direction where it's not diffusive anymore. And this is what we see. So we can actually calculate the characteristic function exactly, although it's hard to invert it and to write the full distribution. But what we can do is look at the marginal distributions, and to see sort of the maximal effect of anisotropy, we choose the directions which are parallel and orthogonal to the initial orientation. And in this case, so if we look at the parallel component, then it turns out that this is again a Gaussian distribution. Okay, Gaussian diffusive. So this is the characteristic function, and this is the scaling form. So it does not depend on dr at all. So the position distribution along the parallel direction parallel to the initial orientation does not depend on the rotational diffusion constant, and it's a Gaussian one. So it spreads diffusively. On the other hand, if we look at the orthogonal component, then it's a very, very, very different behavior. First of all, it shows a ballistic motion. So along the perpendicular direction, the cloud spreads in a ballistic manner, and it has a distribution, I mean it has a scaling form, a ballistic scaling form with the scaling function of product of gamma functions, which is a non-trivial function, and it has exponential tails. So on the one hand, along the direction which is parallel to the initial orientation, we have a Gaussian tail, and along the perpendicular direction we have exponential tails with a ballistic scaling. So this is a new regime which has not been seen for active motion so far as we know. And so it would be interesting to see if it is possible to reproduce it in experiment. Like for Mysococcus xanthus, we have this really well separated time scales, and perhaps it should be visible in the intermediate regime. So let me make a quick summary here. How much time do I have? Okay, I'll be quick. Okay, so basically I have introduced a model for direction reversing active Brownian motion, which is a kind of motion seen in certain bacteria and also some kind of genus particles. And we have seen that the presence of two different time scales and its interplay gives rise to different dynamical regimes, and in particular at an intermediate regime we see a very novel behavior where the distribution scales ballistically in one direction and diffusively along its perpendicular direction. So natural question is now is what happens if we put these particles in an external potential, because it's known that active particles in external potentials show non-trivial non-Boltzmann kind of distributions. It shows crowding at the boundary. So what happens for this case? So in the presence of, we do the first, simplest case first, where we have a harmonic potential of strength mu, and now we have three different time scales: the inherent active time scales, dr inverse, gamma inverse, and the time scale coming from the potential mu inverse. And it turns out that the interplay of these three time scales now also give rise to some new kind of behavior. But before going into that, let me just quickly recap what happens when we have usual active Brownian particles or unensembled particles in the presence of harmonic trap. So this is some simulation for ABP, and this is analytical results for RTP in 1D. So in both cases we see that when the activity is strong, meaning when dr or the flipping rate for RTP is very small, we see that the particles are crowding near the boundary. So they go as far as they can go and they stick there. Okay. On the other hand, when when one increases the rotational diffusion constant, then the particles start to come back to the origin and they crowd near the minimum of the potential. So this is the so-called passive phase, this is the so-called active phase, and there is kind of a shape transition for these usual active particles. So here instead what we have is that we also have these shape transitions, but we see two novel phases, two novel active and passive phases. So here is the phase diagram, schematic phase diagram in the dr gamma mu inverse space. So if what we find is if dr is much larger than mu, so the strength of the trap is weak, and in that case we get the usual passive phase. So single peak near the origin. Okay. On the other hand, if we have this scenario where both the active parameters are much smaller than the trapping parameter, in that case we have the usual active phase, which is this. But on the other hand, if this additional time scale is I mean, depending on whether it's larger or smaller than the reversal rate, then we get two new phases, which we call active 2 and passive 2. So how they differ is that in the presence of an additional peak at the origin. So unlike the passive 1 phase, the usual passive phase, this peak is not Gaussian, it actually has a logarithmic divergence. So that's the way it differs from the usual passive phase for ordinary active particles. On the other hand, there is an active 2 phase where in addition to the crowding at the boundaries, we also have an extra peak at the origin. Okay, this is also not seen for active particles with single time scales. And if we look at the trajectories for these different regimes, we sort of understand how this happens. So in case of, I mean, perhaps the most interesting is active 2. In this case, actually dr is small and gamma is large. So what happens is the particle moves more or less in straight line. So it passes through the origin many times and then it hits the boundary and then rotates a bit and then again does this motion. And this is what gives rise to the peak at the origin. For the passive 2 phase, it's also similar. I mean, it still moves along straight lines, but it doesn't reach the boundary, and before that it comes back to the origin, giving rise to the peak at the origin, but there is no crowding at the boundary, because before it reaches the boundary it reverses its direction. So, so let's focus on this novel active and passive phases, which I mean, just to remind you, it occurs when dr is much smaller than both mu and gamma. And depending on whether mu is greater than gamma or not, we get these two different behaviors. So we could calculate the exact scaling function, which is of this complicated form. But here is a plot showing the marginal distribution. So we see this extra peak, which is there for both passive 2 and active 2 phases, and we see that the boundary crowding disappears when we increase the value of nu, which is the ratio of gamma by mu. Okay, and this peak, I mean, this peak is logarithmic in nature, very different than the diffusive behavior. Okay, so yeah, how much time? Okay, so I'll just mention one more thing. So after we have looked at this single particle behavior, it's of course interesting to ask what happens when these particles are interacting. So we take the simplest possible scenario again. So we consider a chain of this direction reversing particles, harmonic chain, periodic harmonic chain even for the simplest case, and we look at the behavior of the tagged particle. So we tag a particle and look at how the mean square displacement of this tagged particle behaves. So just to remind you, so if we look at a harmonic chain of Brownian particles, then the tagged particle mean square displacement grows as square root t. So here again, for this kind of particles, we expect different kind of dynamical regimes because of this so many time scales, and this is what we get actually. So this is a schematic diagram showing what are the different kind of behavior one can get. So the first column is the very short time regime, where we don't expect to see any effect of the interaction, and indeed we see the TQ behavior, which is the free particle case. And then depending on the order of the time scales, we get t to the power 5 by 2, which is again not seen for any kind of active particles, and then we get t, which is seen for ABP, this pure chain of ABP, and then at very late time square root t, which is reminiscent of the Brownian particle chain as we expect for very long time cases. So yeah, let me just summarize. So I have introduced a very simple model for direction reversing active motion, and it it shows certain interesting behavior, and depending on the relative order of the two time scales, we see the emergence of certain novel dynamical regime. And in particular, in the presence of external potential, we see emergence of different new active and passive phases, where instead of the usual Gaussian behavior, we have logarithmic divergence at the origin. And as I already mentioned, that for Mysococcus xanthus, the time scales are really well separated, and it's actually that gamma inverse is much smaller than dr inverse, and it would be great if this novel phase can be observed in experiments. And another question is, for certain bacteria, it so happens that when it reverses direction, the speed also changes. So the next step would be to incorporate this speed change in the model and see if we can get completely different behavior, or what happens, what is the effect of this. And then finally, these Mysococcus xanthus bacteria, they have certain collective behavior which is called fruiting body formation. So they form a cluster. So it would be interesting to see if we can also get that from this kind of simple models. And let me finish by thanking my co-workers, Sandeep Shahapundit at RRI, and his students Ayon and Shashank. And these are the works the talk is based on. So thank you very much for your attention.
Thank you, Urna. Any questions? Students first, if possible, or postdocs?
I'm waiting for students.
Hi, thanks for the very interesting talk. So if I understand correctly, so this novel phase that you're seeing, this exists in this model, but not in run and tumble particles.
Yeah.
Because it is doing this one-dimensional sort of reversal in this
Yes.
3D space. So in this, so when you are considering interacting particles, so this one-dimensional reversal is only possible if your particle is anisotropic in some sense. Is that right to say? So it's rod-like or?
In anisotropic, yes, but it need not be a, so for example, these titanium oxide particles, they're just coated, half coated with gold, so they don't have any shape anisotropy, but just because the catalytic behavior is different on the two fronts. So what they do there is they shine, so if it moves in a certain direction when UV light is shown, and it moves in the opposite direction when green light is shown. So it's more of a response to the stimuli. It's not in any inherent shape anisotropy or so. On the other hand, the bacteria, they do have some shape anisotropy. So these Mysococcus xanthus, they are rod-like particles, much bigger.
Okay, thanks.
Want to pass it?
I'll go.
So this is might be a bit of I don't know. In the non-trapping case where you're shown, the interesting case of the intermediate 2 where gamma inverse was less than D,
Yeah.
a DR inverse. I also happen to notice that the values of the bacteria are also in the same
Yeah,
regime.
exactly.
So does this confer any advantages in like searching of space or something because of which there is some correlation between?
So we don't know the answer to that. It might just be, so these are bigger particles, right? So it might be harder for them to, because it might be easier for them to move in the opposite direction instead of rotating by a finite amount. But whether there is any evolutionary advantage or any practical advantage, we don't know the answer to that.
I'd like to ask about active 2 and passive 2. Why you call one of them active and the other passive?
This one, right? So we are going by the notion that active particles crowd at the boundary. So here, this active 2, we have this crowding at the boundary. In addition, we have an extra peak. And so these two peaks are same exactly. And in one case, we have this additional crowding at the boundary. In the other case, we don't have the crowding. So this is reminiscent of the behavior of active particles, passive particles, where it goes to the center, to the minimum of the potential. So this is why we call it passive-like phase, although not exactly Boltzmann kind of passive phase, but just passive-like in the sense that the particles are at the minimum of the potential. And in the other case, it also has a huge probability to be at the boundary, which is typical of active particles. That's why we call it active.
Okay. Thank you. Did you derive a generalized fluctuation dissipation relation for this model?
Yeah. Can you repeat it?
Did you derive a generalized fluctuation dissipation relation for this non-equilibrium model to?
We haven't yet.
Okay, you didn't. No.
Hi. Yes, I didn't understand your answer to that question because your...
Which question? Why we call it active 2 and passive 2?
No, the one before.
Shape.
Yes, so these cells that you look at, you say that to change the direction, they don't rotate. What do they look like, these cells? Because if you see
No, they rotate. So let me just go back to the video. So these ones you mean, right? Yeah, so.
Okay, yeah, absolutely.
So what was your question? No, maybe I didn't get it.
I don't know, how are the cilia made so that they can just switch?
Biologically?
Yes.
Oh, that I don't know. But they can move both ways. I mean, they don't have any, they're sort of apolar in that sense. So they can move in both directions.
Okay, very good.
Hi, Urna.
Yeah.
Maybe a bit academic question, but what happens if instead of reversing the velocity, you change the angle by a fixed amount, maybe plus and minus with equal probability? Have you tried?
Yeah, one can try. So actually what happens so if you, so there was a study for the angle change in, for the Putida case, and it was shown that change in angle, the distribution of that is a double peak distribution. So there is one peak near 0 and then another peak near 180 degree.
Okay.
So it's not exactly 180 degree, but it's a narrow peak. So, and one can try with other angles also.
So just
yes.
to understand what happens, for example, in the, I don't know, active phase 2 to the central peak, because at a certain point when you make it continuous, it should disappear.
So if you make continuous what?
The offset in the angle, the discrete offset in the angle.
Yeah, it should disappear, yes.
Okay.
So I expect that there will be, there will still be some novel behavior in this case, but the distributions will be different. I mean, the scaling function will also be different, yeah.
So what if you have an isotropic potential? Does the logarithmic divergence disappear, or do you have multiple divergences?
Anisotropic,
because it looks like this divergence is in the middle because you have basically a...
yeah, so anisotropic potential, if the divergence would completely disappear, I would still expect there would be some crowding at the point, because it's coming from, if you remember this trajectory picture. So here, so this divergence is coming because the reversal is more frequent, and then they are just moving on a straight line. So anisotropic would mean that this boundary would be probably different, but they would still probably reverse the directions. No? So I would expect that the behavior near the boundary would change. There would still be probably crowding, because for ordinary active particles, you still have crowding at the boundary. It doesn't matter what shape is it. What shape it is. And here this extra peak is coming because of the reversal. Okay, so the form would still be anisotropic, but probably there would still be some crowding at the boundary and at the origin still.
Thank you.
Hi, sorry, over here.
Yeah.
I had a question about this intermediate regime where the reversals were fast and the rotation was fairly slow.
Yes.
And so you said that in the parallel direction, it's like a random walk, which makes sense because you sort of take steps and back and forth. But orthogonal, it was supposed to be ballistic, and I didn't quite understand what's the argument for why one might expect that.
What's the
Because it
argument
seems like
for?
if you just had a component, that would also be reversing and it would diffuse.
Yes. So no, I didn't understand the question. So you are asking why, so parallel direction is Gaussian, that's okay, and why is it ballistic in the other direction?
Yeah, like what's the I was trying to pull out the velocity from.
Which velocity?
Well, if it's ballistic, it has a characteristic velocity.
Ballistic, it has a characteristic velocity, yeah, which depends on the ratio of the two time scales.
But is there a more intuitive argument for
why it is ballistic? Mathematically, it's pretty clear why it happens because, I mean, basically, so we see from here, if you look at the mean square displacement expression, so if we look at, so here we are looking at parallel and perpendicular direction, meaning we take theta naught equal to 0. So in this case, there is this t term, which is the diffusive one. And for the y case, this is the diffusive term is actually being cancelled. And we have the next term, which is the ballistic one. So active particles, I mean, typically they are ballistic at short times. And here the effect of the diffusion is somehow being cancelled.
Yeah, because it's just sort of, I mean, naively, I would think, I look at parallel motion that's perpendicular, and you would think that the component is reversed too, and so then it would.
No, that's not, so why are you saying that? So it moves in a certain direction. I mean, initially it was along a certain direction, but then there is also a little bit of diffusion, and then there is also reversal, right? And the reversal basically makes the, I mean, without reversal, if it is not rotating at all, it would be just a straight line, and the reversal is what is making it a little bit, I mean, wider.
Okay.
Right? Yeah, I mean, it's not very satisfactory, but.
Hi, Urna. So I wonder if the model in the active 2 regime can be thought of in the following way, that I have a track on which there is an RTP, and the track itself is changing.
Yeah, that's exactly how we calculate it.
So here's an example. Then could we think of like molecular motor movement in cells on a polymer, and the polymer itself is remodeling. So there's a time scale of the polymer. That's one particular example.
Okay. And can it be seen experimentally?
No, I'm asking, since you asked for
experimental I mean, that's exactly how we derive this analytical expression. So basically, the particles go and, I mean, they go to the boundary and come back, and then once a while, the direction changes a bit, and that change, I mean, eventually over time, that becomes completely uniform. So the direction can be uniform. So that's exactly how you get this.
That's right. No, that could have consequences for cellular transport and so on.
Okay. Yeah.
So when these bacteria swim backwards, right, I think at some point there's a characteristic change in their direction, which I think arises from a mechanical instability of their flagellum.
So for the bacteria?
For the bacteria, indeed. And so have you thought about how that would change the results? Right, I guess you could include.
No, I didn't get the question. What
right, if you also look at these trajectories, right, of these bacteria that swim backwards.
So this is in free space, right? The experimental ones.
Yeah, if you look at these experimental trajectories of these bacteria, right, when they swim backwards. If you look at that movie,
yes.
Right, you see that they constantly have this sort of characteristic change in their direction.
So in this, here or here, where are you looking?
No, here on the right.
Okay,
indeed.
yeah.
And I think that is associated with a mechanical instability of the flagellum,
in this case, no, this is just a Janus particle. This is a titanium oxide particle, and you switch different lights.
Okay.
In this case, there is no, nothing inside the particle. Okay. In this case, possibly, yes.
Yeah. But have you thought about how that, you could include it in a model, right?
That was not our point. I mean, we just wanted to reproduce this motion, just direction reversing, and see what we can, I mean, with this minimal model, what we can get.
Yeah.
Possibly, if we include the more complex features, we will get more complex details, yes.
Yeah, okay.
Also, as I said, I mean, in this case, there is a velocity change with reversal. That also we have not included yet. And here also, if you look more carefully, you can see that for some particles, the whole trajectory is reversed. I mean, it goes like that and then comes back following this thing. So these features are also not included in this preliminary model.
Hear me? Hello?
Yeah.
Yes, so I had a question about the switching mechanism, because you assume that the waiting times are exponential, no? In essence. So I've seen many papers with this assumption. So I wonder, is this a tradition in the field, or is it so my question is there experimental data about these waiting times?
Yes, so for this case, for the Pseudomonas putida case, there is experimental data. And what they showed is that the distribution is more like a gamma function. Okay, so one can try to do that. But usually, the qualitative behavior with gamma function waiting time and exponential waiting time, because the tails are exponential, they are not so very different. I mean, quantitatively, yes, but qualitatively, they are not so very different. But indeed, I mean, it would be interesting to see if one can.
The gamma function has like a peak. So
It has a peak, but I mean, usually not. So deterministically, yes. Yeah. So this probably can be done because in the end, I mean, the calculations that were done were using the trajectory-based approach. So we look at intervals between flipping, and between flipping, we know what happens. It's like ABP behavior. So this can be done, but this is again the simplest possible scenario.
Okay, I'll take the last question. Regarding the bacteria that you see here, do you know anything about the environment they live in? Because, for instance, if they live in a crowded environment, and since they're rod-like, they might get stuck, it sounds like it pays off to have kind of a search mechanism in which you reverse direction.
Maybe. So they actually show this. So it seems experimentalists at least think that this reversal helps in this fruiting body formation. So when the environment is dry, I mean it's devoid of food, so they form kind of a cluster, and then they can survive for long. And this is typical of this Mysococcus xanthus, this fruiting body formation. Possibly it has something to do with this kind of environment and the kind of motion, but yeah.
Okay, let's thank Urna again. And I remind everyone that we have the group photo now, and it will probably be here, and after that,